Um, in terms of, and obviously, like the thing that we want ultimately, and the thing that we are working towards is figuring out ways in which we can keep those threads intact for as long as possible, so that we have many more channels going into the model. That's by far the number one priority that the team is currently embarking on to understand how to prevent that from happening. Um, the thing that I will say also is that, as I mentioned, this is the first time ever that we're putting these threads in a human brain. And human brain, just for size reference, is 10 times that of the monkey brain or the sheep brain. And it's just a very, very different environment. It moves a lot more. It's like actually moved a lot more than we expected when we did Nolan surgery. And it's just a very, very different environment than what we're used to. And this is why we do clinical trial, right? We want to uncover some of these issues and failure modes earlier than later. So in many ways, it's provided us with this enormous amount of data and um, information to be able to solve this. And this is something that Neuralink is extremely good at. Once we have set of clear objective and engineering problem, we have enormous amount of talents across many, many disciplines to be able to come together and fix the problem very, very quickly.
But it sounds like one of the fascinating challenges here is for the system on the decoding side to be adaptable across different timescales. So whether it's movement of threads or different aspects of signal drift, sort of on the software of the human brain, something changing. Like Nolan talks about cursor drift that could be corrected, and there's a whole UX challenge to how to do that. So it sounds like adaptability is like a fundamental property that has to be engineered in.
It is, and I mean, I think, as a company, we're extremely vertically integrated. We make these thin film arrays in our own microfab.
Yeah, there's like you said, built in house. This whole paragraph here from this blog post is pretty gangster. Building the technologies described above has been no small feat. And there's a bunch of links here that I recommend people click on. We constructed in-house microfabrication capabilities to rapidly produce various iterations of thin film arrays that constitute our electrode threads. We created a custom femtosecond laser mill to
Oh,
manufacture components with micro-level precision. I think there's a tweet associated with
that's
this.
a whole thing that we can get into.
Yeah, this okay. What are we looking at here? This thing. This
Yeah.
is so in less than 1 minute, our custom-made femtosecond laser mill cuts this geometry in the tips of our needles. So we're looking at this weirdly shaped needle. The tip is only 10 to 12 microns in width, only slightly larger than the diameter of a red blood cell. The small size allows threads to be inserted with minimal damage to the cortex. Okay, so what's interesting about this geometry? So we're looking at this just geometry of a needle.
Yeah. This is the needle that's engaging with the loops in the thread.
Mm-hmm.
So they're the ones that thread the loop, and then peel it from the silicon backing, and then this is the thing that gets inserted into the tissue, and then this pulls out, leaving the thread.
Mm-hmm.
And this kind of a notch, or the shark tooth that we used to call, is the thing that actually is grasping the loop, and then it's designed in such way such that when you pull out, leaves the loop.
And the robot is controlling this needle.
Correct. So this is actually housed in a cannula, and basically the robot has a lot of the optics that look for where the loop is.